So it's 4 p.m. Let's start now. Good evening, everyone. I'm delighted to welcome our guest speaker of the evening, who is one of India's renowned computational neuroscientists, Professor Upinder S. Bhalla. He has acquired his PhD from California Institute of Technology, and he is an academician and currently a professor in Neurobiology Department at National Center for Biological Sciences, TIFR, Bangalore. He has been awarded with Shanti Swarup Bhatnagar Award from the Council of Scientific and Industrial Research in 2007 for his work in the field of biological sciences. He has also received Infosys Prize in 2017 for his work on the role of olfaction in behavior. So, sir, I welcome you today and thank you for accepting our invitation for the talk. It's a pleasure to have you here with us. Now, I would hand it over to you. And there's a small request to people. You may drop your questions in the chat box and they will all be answered by sir after the talk is over. Thank you.
Thank you, Ruchika. Thank you to the whole club who have been very accommodating. And I really like to thank the audience who have turned out in quite remarkable numbers. I see 100 participants on this Pongal or Shankranti, whatever your favorite holiday of the season may be. And I really appreciate your interest in understanding how the brain works, which is, of course, what has fascinated me all my career. So let me just gear up by starting off the presentation, and then we will dive right in. Okay, are you able to see the presentation?
Yes, sir.
Wonderful. Okay, let me get started. So this is where I work at NCBS, TFR in Bangalore, and I'm going to talk to you about fantastic memories and where to find them. And I'm sure you are well aware of the slightly humorous illusion I have over here. But at least fantastic memories are real and not mythical. And let's spend a little time discussing that. So to start off with, this is your brain, and it's roughly 1.5 liters of tissue. It consumes about as much power as a LED light bulb, about 20 watts, which is astonishingly little for what it does. And the point I'd like to emphasize, and hopefully will come across in this talk, is that to understand its functioning, you really have to think of it as an integrated system, which is operating at all of the levels indicated in this diagram, all the way from molecules, synapses, neurons, and subcellular portions, circuits, systems, all the way up to behavior, which is the result of its activity. So these are all levels of computation of the brain, and although there is a tendency to do so, it is not a good idea to restrict yourself to just one level, because computation occurs seamlessly across all of these levels. And I'll try and give you a glimpse of that. So let's just run through the scales. The brain, as I said, is about 1.4 or so liters, about 10-15 centimeters across in this axis. And if you cast your eye towards this corner here, you will see a little dot, which is roughly the size of a neuron. So for comparison, this is the brain, and this is roughly the size of a neuron if it were next to it. So now we jump in scales. We're going to now expand so that that entire field of view is one neuron. This is actually a model of a neuron called the Purkinje neuron, and this model was generated by my colleague Eric de Schutter many years ago. So this is around 0.5 millimeter across. Yeah, and as you can see, it's a lovely, beautiful branching structure, and indeed, all of the neurons in the brain have their own distinctive, often very elegant branching structures. But let's look at the scales one more time. If you now zoom in onto this little block here of about 0.5 micron, 1/100th of this, Let's see what we get. And this has indeed been done through very, very painstaking electron microscopy, serial sectioning, to reconstruct a small piece of dendrite from that neuron, and that is indicated in yellow. Running across this small 5 micron cube of brain tissue are lots and lots and lots of axons, of which one is particularly clearly indicated over here. And we can home in still further. The little red dots that you see are the contact points between axons and this dendrite. And these are, of course, as you all know, called synapses. And these are roughly 0.5 micron across. And there's thousands of synapses, even in this tiny volume, extremely densely packed. And I think one of the key things you should take away from a diagram like this is to understand that the brain is not your sort of spread out, occasionally here a neuron, occasionally there a neuron. It is utterly densely packed. Every cubic micron of it is full of circuitry, contacts, parts of neurons, and so on. It is a very, very dense system, and which is why you can pack so much computation into such a small volume. But let's zoom in still further. Now we're talking about the size of a single synapse. And this structure here, which is beautifully visualized by Sven Truckenbrock, who gave you this diagram, is the presynaptic bouton. And the postsynaptic bouton, postsynaptic spine is roughly the same size, about 0.5 micron across. And now we're zoomed in close enough that you can start to see the incredible tangle of protein molecules, filaments, globular ones. You have some microtubule structures here, lots of vesicles floating around. Right down here, I don't know if you can see my mouse cursor. Right down here, you have the synaptic release zone. So I just really want to stress to you that even at this scale, the molecules are incredibly densely packed. So it is, and these are the ones which are doing all the computation. So we are really talking about an incredibly compact structure, utterly packed with computational elements. And at this scale, we're talking about the individual molecules and proteins. And we will have a discussion on these as well. Okay, so let's step back now. Let's start at the sort of the highest level, the behavioral level. And this is something which you are almost certainly all totally familiar with, which is the work of Ivan Pavlov, a minute Russian scientist, who actually was looking at digestion. He's looking at the enzymes in saliva. And for that, he had this device which measures salivary flow. But he was an observant fellow. And you all know the story that every day at a certain time, there would be the delivery of food. And of course, the dogs, when they saw the food, would salivate. That's the unconditioned response, the salivation. And the unconditioned stimulus is the food. And because it was a well-ordered lab, there would be a bell to tell the staff, okay, now it's time for food. And initially the dogs did what they do when they hear a bell, which is nothing very much. But after a while, and this is where a good scientist's observation comes in, he observed that the dogs started salivating at the time the bell rang. So basically enough associations between the bell and the food led to the dog salivating at the time of what was originally a neutral stimulus. The bell has nothing to do with food in the normal life of a dog. So now the bell, and in fact, you could get the dog to salivate with just the bell alone, without any food. And so he analyzed this in great depth, and a great deal of work has followed since. And this is the classical conditioning. That's in fact the name of this protocol, where you combine a neutral with an unconditioned stimulus, and eventually the animal learns to respond to the neutral stimulus after enough associations have happened. So what I'm going to do now is I'm going to do a little exercise to give you a sense of how you could implement this. Of course, this is a very cartoon diagram, right? It's a cartoon dog. It's a cartoon dog with 3 neurons. Yeah. What we're going to do in this cartoon dog with 3 neurons is to look at how you can achieve such kinds of conditioning through the functioning of neurons and the functioning of synapses, which can change their weight. And I will explain all of these terms in a moment. But let's start with our cartoon dog. So our cartoon dog has 3 neurons. The first neuron of the cartoon dog is this green one over here, which responds to food. And this one is an unconditioned stimulus and response. So in other words, when food comes, the neuron fires and that causes saliva to be released. Very, very straightforward. There's another cartoon neuron here. This is for the bell. This same thing, when the bell is present, this neuron fires. It connects onto the food neuron, but it's a weak connection initially. And so nothing much happens. Similarly, there could be yet another neuron which responds to, let's say, a certain colored light. That's the pink one over here. And yeah, initially the light goes on. The dog's not terribly interested in it. It has nothing to excite it about food or anything. But now we proceed with the associations. And so here's our experimental condition that the bell is always associated with the light, with the food. And here's a control, that is the light is not associated with the food. And what happens is that after enough association, various chemical processes, biochemical processes, and electrical processes take place in the pre- and postsynaptic cell at the synapse. And what used to be a weak synapse from the bell to the food neuron becomes eventually a strong synapse. I'm going to call it a switched on synapse for reasons that will become clearer much later in the talk. And so once this association has been strengthened, once the pre- and postsynaptic association, the presynaptic event and postsynaptic event have been associated enough time, in other words, the conditioned and the unconditioned stimulus have been associated, this synapse becomes stronger. This is called plasticity. It becomes stronger, and the effect of that is that now when the bell neuron goes off, when the bell happens, the bell neuron is active, the synapse is now strong enough to trigger the food neuron, and this triggers salivation. And so in this cartoon animal, you have learned association. Pavlovian conditioning has happened. And of course, the control neuron has not been seen association, so its synapse remains weak. So here is a cartoon version of classical conditioning. And the reason why I present this to you is that you should be able to see, even in this cartoon form, how you can relate behavior at the very top of that hierarchy to cellular events and all the way down to synaptic events. And of course, what's happening at synaptic events are in fact chemical, molecular events. So all of these things are related when it comes to learning and behavior. Okay, so by the way, anyone who wishes to ask a question as we go along, it should be a short clarification question, is most welcome to do so, and I'll be happy to answer. All right, so if there are no questions at this point, I will proceed. So now let's go into the brain a little bit. And here we have a rather sad, tragic figure by the name of Henry Molaison, who for many decades was known as patient HM. And why patient HM was famous was because he had intractable epilepsy, which led to surgical intervention. And a region of the brain called the hippocampus, which is over here, I'll show you another picture of the hippocampus in a little while, it's over here. This was completely removed on both sides, bilateral removal of hippocampus. Yeah? And he recovered from the epilepsy, but he lost his ability to form new memories. And this was kind of interesting. He retained his old memories. So if you asked him he was American, so who was the US president when he went into the hospital, he knew that. He could tell you, for example, his favorite players in the baseball team. So he had all these memories, but he could not form new ones. And in fact, he always lived his life, so to speak, in the past. And of course, this led to considerable distress as he got older. So he lost the ability to form new and short-term memories, but he had long-term memory was intact. He remembered existing memory that had already been stored. And interestingly, he still retained the ability to form procedural memories. And procedural memories are the kinds of things where for example, when you learn to ride a bike, or in his case, they tested by learning how to draw something in a mirror image. So he was able to learn these things, even though he had no recollection of actually having done the task. And there were other interesting things they looked at, which I won't get into for lack of time. But the point is that here, again, we're looking at memory and where certain kinds of memory are located in the brain. And this formation of new memories and short-term memories is, by this set of experiments and many that follow, very, very strongly associated with this region called the hippocampus. So that's another interesting place to look for memory. So let me show you a little bit more about the hippocampus. I hope you can see this rotating head, semi-transparent, in which there are these little red lobes, kind of banana shaped. Yeah, you can see this?
Yes, sir.
Good. Okay, so those are your hippocampuses, left and right hippocampus. And this is where a great deal of short-term declarative memory, as they say, is being stored. So if you happen to remember any of the facts or ideas that are put forth in this talk, this is where it is going for the short term, and then in the long term, it may go to other regions of the brain. Now, the hippocampus is also important for many other kinds of memory. So this is one of the most famous kinds, for which the Nobel Prize went to the Mosers a few years ago, and I think to John O'Keefe as well. So this is the formation of what are called place cells, which are cells in your hippocampus and the rat hippocampus, which respond when you are in a certain place. That's why they're called place cells. So if the animal is running along, let's start with this kind of semi-one-dimensional shape. If it's running along on this linear track, there will be this neuron. Let's indicate its activity by green dots. So this neuron will be active only in this zone. And maybe this other neuron over here in blue dots will be active only in this zone. And the interesting thing is that the animal has only to experience this location very few times, once or twice, and it forms these representations. These neurons start firing in those locations, and then they stay. So anytime it comes back to that location, those neurons, the same set of neurons, are active in those same locations. This also works in 2 dimensions. So here in red is indicated where the neuron is active when it's running around in this arena over here. And you can see it's running all over the place, but it's active only here. And this is kind of a heat map to show where it is active. So this is, again, a form of memory. And this, again, is happening in the hippocampus. So let's now talk about a little bit of fantastic memories. All of these are really fantastic, but here's an illustration of just how fantastic it can get. So I'm citing a book by the name of Mind of a Mnemonist. This is an old book, 50 years old, published, written by Alexander Luria, who's a great Russian neuropsychiatrist. And he had a patient whose name is not told, which is the correct thing to do until the patient passes away. And this patient had, as far as one could see, perfect memory. He could remember everything. And before you start thinking, oh my, this would be a wonderful thing, I would pass all my exams. Actually, this was a patient, mind you, yeah? And the reason he was a patient was because he actually had a pretty miserable life of it. In the end, he was reduced to, or elevated to, depending on how you look at it, being a performer as a sideshow in a circus. Why is this? So it turns out that the capacity for almost arbitrary recall was accompanied by a huge difficulty in discerning features, patterns in observations, in stimuli. So here's an example. So this patient was given any number of tables of numbers, and he could recall them 10 years later, kind of thing. It was mind-boggling, all carefully noted by Dr. Luria. Here's an example of one of the tables, though. 1, 2, 3, 4, 2, 3, 4, 5, 3, 4, 5, 6, 4, 5, 6, 7. Yeah? Okay, now you and I look at this table, and we immediately see that there is some structure to this. Yeah? You don't really need to remember all the individual digits. But that is exactly what this patient did. He did not perceive the pattern. He just remembered it. And 4 or 5 years later, he just recalled it. And then when it was pointed out to him, he said, oh yeah, they are in order. I didn't notice. Okay, so maybe you get a sense of what are the deficits that come when you have a fantastic memory. And there are many other absolutely fascinating things. Luria was so meticulous. He recorded all of these things and what the patient was experiencing and thinking. And it was clear that this patient was a synesthete, which means that he tended to mix different kinds of stimuli. So for example, certain numbers gave him the perception of certain colors or textures or sounds. And that helped him to recall these things in a manner which is still somewhat mysterious, because he did this, he could remember random tables for decades. It was amazing. In fact, his problem was to forget things. Okay, so this is an example of a fantastic memory, though as I said, all memories are actually quite fantastic given what they can do. So now let me just quickly go over this again and sort of recap some of the levels of memory where one can look for them. So at the systems level, when you're talking about large brain regions, two particular regions come to mind. The hippocampus is great for declarative memory. We've discussed some examples of that. And there's a small region of the brain called the amygdala, which is crucial for emotional memory. So for example, stress or even happy emotions, those associations seem to have a great deal of reliance on the amygdala. For example, if in people who are exposed to traumatic events and end up with post-traumatic stress disorder, the amygdala is a key player in that. And my colleague Shona Chatterjee here at NCBS has worked on this for many years. So that's a systems level view. The area level view, and we'll see a bit more of this, is there are certain kinds of circuits which are very good for storing information. At the cell and circuit level, the connectivity changes, that's the synaptic plasticity I told you about, but the excitability of neurons can change, and even the gene expression can change. If you go down even further to the synapse level, the conductance of the ion channels, of the receptor ion channels starts to change. The location of the channels changes, the amount of neurotransmitter changes, and all of these are components of a memory. So when you look for memory, you need to look at all of these levels. They're all part of the whole. You cannot blinker yourself and look at any one level. You have to look at the entire system, at the cells, at the synapses, at the subcellular regions, and at the synapses and molecules. Yeah. So I'm going to spend a little time talking about just a little glimpse of some of our work on memory, and this is at the systems level. So we are using a technique called 2-photon imaging of a part of the hippocampus called the CA1. So here's a mouse or rat hippocampus, this banana-shaped structure, and it's a relatively deep region. So we need to use, of course, make a small hole in the skull, which we cover up with a glass window. And we need to use a technique called 2-photon imaging, which allows us to image the activity of cells using various clever genetic tricks quite deep into the brain, a millimeter or so, as much as a millimeter. So this is what we're doing. Here is a picture of the metal plate that we put on top, which has got a glass opening, a glass window into the skull. And there you can just about see the fluorescent region of the brain over there. And here, after the experiment is over, when we've sacrificed the animal, is the fluorescent region of the hippocampus in bright color. So this was using a dye. You can use genetically encoded reporters. I won't go into those details. There's some absolutely marvelous techniques out there in neuroscience now, which allow us to do these things. Here is just for those of you who like techie things, here is our a custom-built 2-photon microscope. The old one was called Bhalla Fry, and the new one is called the Black Widow. And don't ask me, this is a choice that the lab decided that this thing looked like a Black Widow spider. I'm sure that they were only thinking of spiders when they were discussing this. Okay, it does look vaguely like a spider with these long legs and struts that you see over here. And here's an image that one can get from such a microscope. And this is training that we do. So of course, we're trying to study memory, so we train the animal. This is extremely similar to Pavlovian conditioning. It's called trace conditioning. So there's about a half-second gap between the conditioned stimulus, which in this case is a sound or a light, and the unconditioned stimulus, which is an air puff to the eye, which causes the animal to blink. So it's very easy to measure, very precise timing, much more precise than salivation. And we can train the mouse quite quickly on this kind of thing. And so here's the sound or light stimulus. There's the air puff. And the animal has to remember this association after a few trials. And here is one of the first images we got from our old microscope from Bhalla Fry. And I don't know if you can see, I'm still really excited by this, but the little dots, which are cells, are blinking on and off. And what you're seeing literally is the activity of the brain in an animal which is experiencing some sensation and remembering some of it. That's why it's so cool. And of course, you have to do a huge amount of analysis to make sense of it. But this is old recording now. It's 7-8 years old, but I still like it very much. Okay, so one of the kinds of things we study, and I'll only give you a very brief glimpse, is the life cycle of a memory trace in the hippocampus. How is it formed? How does it compete with the different kinds of information? How can different kinds of information coexist? How can you remember more than one thing at a time? And how do you forget things? So these are all part of our research program. I don't have time to go into much depth on any of these. And so just to give you a glimpse through of the first one, formation. So what we found, and this is a work by Mehrab Modi and Ashish Thavle, as I said, about 8 years ago now. And what they found was that before learning, just to orient you, here are 6 neurons that are recorded. For each neuron, we're showing 3 trials, so you can just see that this is a consistent effect. So 6 neurons, 3 trials, and each trial at some point has the CS, conditioned stimulus here in yellow, and the unconditioned stimulus, the air puff over here. And initially you can see there's activity all over the place, not much happening, not very strong activity either. The little asterisks indicate the peak activity for that particular trial. It's all over the place. After learning, which is just half an hour, 45 minutes later, a few trials later, some 20, 30 trials later, suddenly something interesting has happened. And we see that the activity of the cells starts to line up. That is, cell number 1 starts to fire, starts to be active just about the time the CS comes in. Cell number 2 is active a little bit later, 3 is a bit later still, and so on. And this is reliable. It happens trial after trial after trial. Here is the formation of a memory, which you can see through behavior, and you can also see through the activity of the brain. And the interesting scene is how you can form a memory and what is happening in the brain when you form that memory. And what's happening is that the activity of cells becomes stronger and it becomes organized according to the different kinds of stimuli. And importantly here, it is bridging the interval. So we have cells which are not active at all, which are in red. We have cells which respond to sound. We have cells which respond to light in dark blue. And then we have cells which respond to both. And this is how you can have cells that are, you can have one region in the brain which can store and respond to multiple memories. And so this is how you can have coexistence of memories. So I won't go over all of these, just to give you a flavor of the kinds of work that one can do trying to understand the neural basis for memory. Okay, so now let me just dive right down to the molecular level and start to look for memory at the level of molecular circuits. And you remember this slide. This is the presynaptic side. The postsynaptic side is equally complicated and marvelous. So here's a model I made of some of the chemical pathways in the postsynaptic side. And I made this a long time ago, 20 plus years ago, when I was a postdoc, just to give you a sense of the complexity. And this is only a tiny fraction of the kinds of molecules that are present, of different proteins and their interactions. What you can do, for example, in these models is you can analyze how signals propagate, and you can analyze whether the propagation will be successful if the signal is in order. That's on the left. So this is an ordered input sequence. And if you have a disordered input sequence, you'll find that the response is very weak. It dies down after a very short time. So look over here, this input is jumping around. And it turns out that this is not strong. So this is just an illustration of some of the kinds of computations and the kinds of models that we carry out to analyze brain function. And so now let me get back, remind you about this particular example of the Pavlovian learning. And let's look a little bit at the molecules that can do this. So some of you may be familiar with the concept of bistability, which is a dynamical system that can exist in 2 different states. And this is often drawn as a kind of energy. Here we have a dynamical system, just an example kind of thing. And you can think of it as 2 distinct energy wells. And your system can exist either in one state, state 1, or if it's given a strong enough push, it can roll down to state 2. And the point is that either of these states is stable. And now we think of how you can have a chemical system adopt to different states. And why do we want this system to adopt multiple states? The idea is that if the synapse can exist in state 1, which could be a weak connection, or state 2, which could be a strong connection, then by switching from one state to another, this synapse could store information. It could undergo plasticity of the kind that you saw in this Pavlovian conditioning model. So let's see how we can do this. So let's take a set of molecules A and different set of molecules B, which happen to interact with each other in some kind of a loop. Yeah. So here's A and B as individual molecules, as groups of molecules, and here they are as the individual molecules of type A and B. So someone has raised their hand. I'm willing to answer a question if it is a serious one. No? Okay, I will just carry on then. All right, so here's A and B. They're in a feedback loop. The same A and B, now think of it this way. Supposing I give a strong stimulus to A, so that's the dark blue over here. So this strong stimulus will cause, because A is turning on B, it causes B to become active. And because there's a feedback process indicated by this line, A is now reinforced, its activity continues. And so you can have the system where both A and B are quiet, and you can think of that as state one. Or you can have the same system, once it's been activated, once it's received a strong push over this energy barrier, it can end up in state two, where both of them are highly active, and all of the individual molecules in this synapse, say, are active. So why is this important? Well, now let's sort of embed these molecules A and B, the triangles and the circles, in the synapse. And this is sort of the gray region sort of meant to roughly represent what's called the dendritic spine. And so this is fine. They can be active or inactive. They can store memories. But the problem here is that molecules in the brain and in the rest of your body turn over, but they really turn over very fast in the brain. So the lifetime of a typical molecule is a few minutes to a few days. That's not very good for storing information for a long time. How can you do it? So here I'm indicating by the crosses the loss of these molecules, which were part of this switch, which was storing information. So now the molecules have been lost. There are gaps in this set of molecules. But of course, the cell is alive, so it synthesizes new molecules, which are indicated in pale blue. Now, the crucial thing is that the new molecules, they do not, quote unquote, know, they do not have any idea what was the state of the synapse before. They're fresh molecules, right? They're just being brought in. So, but what happens is that because the vast majority of the molecules here are still in the active state or the inactive state, by their activity, for example, these can turn on the new molecules. And in due course, you will have turnover of other molecules, and more new molecules will come in. And in this kind of feedback process where new molecules come in, are brought into the same state, and old molecules can die, you end up with being able to store the information for much longer than the molecules themselves last. And this is how, in principle, one can store information for 100 years, the lifetime that we should all aspire to, or maybe more. Yeah, even though the molecules involved last maybe a week. So by this feedback, turnover, bringing to the appropriate state, and then carrying on. So this is one reason why it's very important to study the molecules. It addresses the problem of how you can store information for a long time, even though the molecules themselves are being lost. And of course, it's always much more complicated than this. There are many more levels of events that take place. But this is, I think, a very powerful concept about how you can store information for a long time. There are other problems with storing information in synapses. Here's one which I spent many years looking at, which is the problem of noise. If you have molecules in a very small volume, such as a synapse, you can't treat the reactions as your standard mass action A plus B gives C kind of situation, because that is a deterministic. That assumes a vast number of molecules. It assumes that you can use floating point numbers, real numbers, to do this. But actually, molecules are integers. There's finite individual numbers of them. And so you can't use differential equations. You have to use stochastic calculations, which basically means that every time you have a couple of molecules that bump into each other, there's a certain probability that they will go forward and form molecule C, and then converse probability that they'll just bounce off each other and remain as individuals A and B. Now, this is a problem because the smaller you get, the fewer molecules there are, the more noisy it becomes. How do you store information in a very noisy environment. So we analyze this a lot. For example, here's a typical feedback loop that we analyzed, which can store information if you make the large volume assumption. But this very same feedback loop completely falls to pieces when you take it to small volumes indicated in the two blue curves. So the red curve is what you would have if you used differential equations, if you use large volumes. It looks nice, yeah? You give a stimulus and it turns on and it will store that information. But the blue curves are terrible, both of them. So these are different runs of the same curve. Here you have this spontaneously turning on, this one spontaneously turns off, spontaneously turns on again, this other one spontaneously turns off. It's a miserable way to store information. You don't want to use these things. So how can you store information for a long time in these synapses? It turns out, again, as I said, I worked on this for a long time. It turns out that certain kinds of combinations of molecules, especially those which involve structural change, especially those which involve certain kinds of higher concentrations or certain kinds of scaffolded multi-step reactions are capable of storing information for years. In fact, we could run the simulation for 100 years, which, as I said, is a good target lifetime for storing information. So this addresses another molecular level question of how you can store information for long times. So I've tried now to give you a glimpse of many levels of computation that the brain undertakes. I'm now going to jump away from real brains to artificial brains, or I should really say artificial neural networks, because this is something which I'm sure you've all heard a lot of interesting stuff about, and it is very interesting. So I'm going to just give you a glimpse of one kind of artificial neural network. It's a very vast field. I can't possibly cover all the interesting topics here, but let me just give you a glimpse. So what are artificial neural networks? So they get their name, as you might expect, by originally, well over 50 years ago, being thought of as an approximation to how real neural networks could function. So they're approximations, they're unrealistic, but that doesn't mean that they don't tell us something. They can be very, very informative. And it has turned out, much to everyone's surprise, that they're actually very, very useful in the real world. One of the fundamental things that they can do very well is to classify things. If you give them lots of information and you train them in certain ways, and I'll tell you a bit about that, they can classify things very effectively. And a crucial thing here, which is a link back to the biology maybe, is that here too, it is the connection between these artificial neurons. It's the synapses, so to speak, which store the information. The weights, the strength of those synapses is important for storing information. And another crucial, very interesting thing here is that these networks, again, quite a lot like real biological networks, learn by example. And in this, they differ from your conventional computer memories, where if you want to, for example, store something, you actually have to explicitly write everything down like a file and store that file. You can't say, no, I don't want it this way, I want the font size bigger. You can't give it an example, say, I want it to look like this. But that's what these neural networks can do. And let me just give you some examples of this. First thing, what are the neurons in this artificial neural network? What are they? They're very, very simple objects, and most of you are probably familiar with this. You have multiple inputs. We're treating the activity of these inputs as some analog variable. And this analog variable is multiplied by a weight, a synaptic, quote unquote synaptic weight for that input. And you get multiple such inputs. And these are then they're summed. So you have a whole bunch of inputs, each has its weight, so you get a weighted sum. And then there's typically some kind of a nonlinear output function, which then gives the output, which can then become the input to the next layer. Yeah, very, very, very simple thing. And yet you can do amazing things with these kinds of abstract neuron models. You can layer them into multiple layers. This is a typical feed-forward artificial neural network with 3 layers. It's got an input layer, a hidden layer, and an output layer. And this is an architecture that you'll see in a moment really works astonishingly well, even for big, big, big networks. Here's a very, very old example of what one could do with this. As you all know, English has no particular logic to its spelling. You pretty much have to learn how to pronounce every word. There's some hints there in the letters that are used, but the input letters map in a very complicated, irregular way to the phonemes, to the sounds that are made. So the word neuroscience could be input to this network. And after a lot of training, this was work by Terry Sichnowski and his colleagues, after a lot of training, it learned to pronounce the word neuroscience properly. And there are many interesting things about this. For example, during the learning process, he coupled it to a speaker. And it sounded like a child learning to babble. It's a child going blah, 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 na, 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 na. So it was making similar kinds of odd, repetitive sounds, a lot like a child does. I don't know if that is suggestive or not about how the brain does it. But you have to be a little cautious. I'll just give you this little story about neural networks. So one of the unfortunate things about many technologies is that very often people decide that this would be a wonderful way to fight, to wage war, to kill other people. And so not surprisingly and depressingly enough, this is exactly what has happened with AIs and even with this very old 20-year-old neural network example. So in this case, this was related to me by a fellow graduate student. There was some department decided that they wanted a neural network that could identify tanks in an image. So you could take satellite pictures and you could say, oh, here are the tanks. Now, of course, we can do it very easily. This was in the early days. So what did they do? They sent a soldier out. He took a lot of pictures of tanks from different angles, and he fed them into the neural network. But of course, you have to also have non-tanks in your data set. So they said, aha, we need non-tanks as well, which is everything else in the universe. So then they sent this chap out again, and he took a lot of pictures of things which were not tanks in the world. And so then you train the neural network, and this is one of the key things, right? You train the neural network by example. You say, you give it an image, and then someone at the other end is saying, and it's generating an answer, and you tell it, no, this was a tank, or this was not a tank. And you do this again and again and again. And by example, the network starts to adjust to synaptic weights. I won't go into the details of that. They use a technique called backpropagation. And after a lot of training, this network learned to recognize tanks. It could classify tanks in the training set, and it could classify non-tanks. Beautiful. So they said, okay, now let's try it out with some new images. And it failed utterly. And they went back. Why was it working perfectly with the training set and not with the test set? And they realized that the first day the soldier had gone out, he'd taken a picture. It was a cloudy day. So all the tanks had been photographed under cloudy conditions. And the next time he went out for non-tanks, it was a sunny day. And so what the neural network had very sensibly done was, I can classify this perfectly if I look for whether it is cloudy or sunny. And so this is an example of an excellent classifier, but it didn't quite give what you wanted. So you have to be careful. Neural networks are very good at classifying things, but you have to train them well. You have to give them a good, diverse training set. Otherwise, they can give you all sorts of funny results, as I'm sure you all know from stories in the media. So now we go many, many, many years later, decades later, and we are in the region of deep learning. And remarkably, this is essentially the same idea. You know, it was 2 layers back in the 70s. Back when I was a graduate student, it was a 3-layer network. Now it is tens, decades of layers. It's huge numbers of layers, millions and billions and trillions of synapses. The reason why this has become possible is twofold. One is that the computational power has gone up enormously. And the other is that the amount of data has gone up enormously, thanks or otherwise to companies like Google, which consume data all the time in vast amounts. And so the same algorithm of backpropagation with some refinements is still used, and it can do these amazing things. And let me just give you some fun things that you can do with deep learning. So first of all, you know that they can talk to you. Those of you who have the misfortune of owning some of these devices will find that they talk to you and listen to you particularly when you don't want them to. But I'll give you some examples here. They can imitate Shakespeare. I'll show you an example of that. They can recognize you in a crowd. That's a little bit worrying. And of course, their primary goal is to sell you things. So you can look at this at your leisure. Those of you who haven't seen XKCD, I strongly recommend this one, which indicates what was possible for a neural network at different stages in human history. So I remember vividly the first time a neural network beat a human being, or a computer beat a human being at chess. That was a big shock to my system. Now we sort of take it for granted, and now Go and all these things are Jeopardy. All of these are done better by computers than by humans. But look at this table. It's kind of a fun thing. The key point here is that no matter what threshold we set, computers have always with time been able to exceed it. So let's see where they get to in your lifetime. But here's some fun stuff. Here is a neural network that was trained on Shakespeare, collected works of Shakespeare. And it is now asked to generate something that looks like Shakespeare. And those of you who've read Shakespeare, this looks remarkably like Shakespeare, even though the content is complete garbage, right? If you read it, it doesn't make sense. But the word structure, the names, even the iambic pentameter, which is the rhythm in which Shakespeare wrote, that's all there. You can do crazy things like this. This looks like perfectly reasonable math. It's actually generated from LaTeX. And so what they did was to feed this pure neural network a huge number of theses, which had lots of LaTeX text in it. And it comes up with the stuff that at first glance looks totally reasonable. Yeah, astonishing what it can do. You could feed it the entire source code of the Linux kernel, which is now 1 million or so lines, and it can generate code that looks like code. Astonishing. I don't know if it would compile, but it's remarkable that it can generate something that looks like this. Here's another example. You can use retinal scans and they've been used for a long while to identify diabetic retinopathy. In other words, damage to the retina caused by diabetes. This makes sense. It's a sensible thing to do with retinal scans. But Google, of course, they also managed to get all the other associated patient data for these patients. And what they discovered was that using exactly the same retinal scans, they could do the following. They could identify the age of the patient within about 3 years. They could almost perfectly identify the gender of the patient. They could tell whether the patient was a smoker. They could tell the blood type. They could tell the blood pressure. And they could tell whether this person had had a heart attack just by looking at the retina. So these are some of the things that a neural network can do. And I don't know if you've seen this one. Yeah, this is through a slightly more elaborate thing called adversarial neural networks. I won't go into the detail. But these are real faces on top. Yeah. And these are kind of stimulus faces on the left. And everything over here, all the other faces are computer generated. They're AI generated faces. And they look astonishingly real. Yeah. I mean, the amazing thing is that, for example, here, this face has no mustache. This face has no mustache. But this neural network has come along and put in a mustache that actually looks pretty reasonable. So anyway, these are some of the fun things you can do. And it all comes down to learning synaptic weights. How does the network learn what are the patterns of faces in the world? And then it adjusts all its weights in order to be able to store that information and recreate them. Okay, so why don't I now start to wrap up a little bit? Yeah, where does one look for memory? And I think I've tried to make the case that you look for memory at all levels of brain function. Yeah, at the behavioral level, the cell level, the subcellular level, even the molecular level. I've given you some examples of the kinds of experiments we do. We do recordings from brains to watch how the activity changes when the animal learns. We do simulations of different kinds. Here's a cartoon model of some of this. We do models of how molecules interact to store information at a very, very basic level. And we try to put all of this together to better understand memory formation. And I'll just wrap up with this thought, a few thoughts about artificial intelligence and brains and memory. So David Marr, many years ago, said that there are 3 levels at which one should try to understand brain function. One is computation. What is the kind of task that the system is trying to solve? What is the computation? The second is the algorithm. What is the concept behind the accomplishment of this task? And the third is the implementation. And the implementation could be in your brain, or in principle, it could be on a computer. So this is a very influential way of thinking about it. And so you could argue that maybe the algorithms that we are using are similar to the algorithms that AIs are using. It's just that we are implementing it in a very small and very compact and very energy efficient volume here, whereas the AIs take gigantic server farms to do. It seems astonishingly, to me at least, that these vast machines, which are basically doing weight lookups, are able to extract the essence of so many kinds of problems. That is remarkable to me. It cannot be distilled in this way. And of course, but now, when you think about it that way, maybe the way you think about the brain doing it is that the brain is much more efficient, much more compact at performing this encoding of all this diverse stimuli in the universe. And a lot of the reason why it can do this is because it has had millions of years to evolve to do this efficiently and maybe to map very, very specifically to certain kinds of problems that come up frequently in the real world. Here's one of the big remaining questions in AI, which is the question of general intelligence. How is it that I can learn, some kind of math and then be able to solve other math problems? How is it that I can learn, even a simple thing in robotics, like being able to walk and then being able to walk on top of some obstacles? This is generalization, and the field is called artificial general intelligence, and it's been surprisingly difficult to achieve. So I suggest that evolution has given us very efficient ways of seeing the underlying patterns in many, many domains of the real world, which can be sensory, motor, social. Social interactions are hugely important for us as individuals and as a species. Real world physics, all of these are encoded, and perhaps there's some bonus encoding that has come to us through our genes. We're very good at this because we need it to survive. And I speculate that at some point, the artificial intelligence will eventually be able to get to the point where it can do some of these things, but it will take a long time before you can do it in that kind of a volume. Okay, so I'd like to thank all of you for your patience in listening to me. I hope I've been clear enough and given you some stimulating ideas. I'd like to thank the many people who've been associated with my lab over the years and the various people who funded me. And thank you to the club, which has allowed me to speak to all of you. Thank you. I'd be happy to take questions.
Thank you, sir, for giving this wonderful lecture and coming here at the first place. And so there are some questions, and some people have also raised their hand. So can we ask them to unmute to ask question, or shall I read out their questions if possible?
Whatever you prefer. You're the boss.
Okay. So there is already a question in the chat box. The question is, memories are stored in the hippocampus and amygdala and many other levels, but a central input from the environment must be present for a human to generate the required chemical process to generate the memories. Does that mean a human with complete loss of senses, all five, can't have memories?
Well, that's not, you know, of course, that you can store memories of things you've thought of. That's actually a very important part of memory, right? So even when you're lying in your bed, you can shut your eyes and it can be nice and quiet and you can still think of things and remember them. So memories are just ways of modifying synapses, modifying activity based on other activity. The source of the activity can be whatever else. Yeah. Of course, I have to say that someone without any senses at all would be in a pretty bad way and may have a lot of other strong problems to contend with, and learning new things would be perhaps one of the least of them. I hope that answers your question.
So there is another question. The next question is, in context of Pavlovian conditioning, what tells the brain during the training to change or modify the structures to establish the association, like strengthening synaptic connections?
Sorry, I didn't quite get that. What is the brain doing?
What tells the brain during the training to change or modify the structures to establish association, like the strengthen synaptic connections?
Okay, so I didn't get into the mechanisms of it, but it's actually something that's been worked out many years ago. Yeah. So when you have activity which is associated across two neurons, you have presynaptic activity and postsynaptic activity coincident. Then there are certain ion channels, the NMDA receptor is one of them, which will open only when it gets presynaptic input, in other words, it gets neurotransmitter, at the same time as there is postsynaptic activity. In other words, when there's a coincidence of the input and the output. In other words, there is an association. So here you have in a molecular level the capability to form associations. And once that is done, then you can trigger many molecular pathways, second messenger pathways, to strengthen that particular synapse. So the mechanisms for doing this level of association are actually reasonably well understood.
There are two more questions. I have asked them to ask in the chat. So I think we will wait a little bit more to see if there are other questions.
Sure thing.
I have a question.
Yeah.
Sir, when you were talking about those stochastic modeling of molecules, so how do they affect this memory building? Can you a little bit explain?
So here's an example. Yeah. So here is the deterministic model is in red. So this is what would happen if you had huge numbers of molecules, or if molecules were continuous variable, right? And in blue, in the two cases in blue, we have examples of what happens when you are dealing with finite numbers, small numbers of molecules. And the fundamental issue is that reactions ultimately occur through individual molecular collisions. And the outcome of these is a probabilistic matter. And therefore, you end up with what looks like noise, and it is in fact related to the temperature. You have something that looks like noise, and so your nice bistable switches, your reactions that are in feedback loops, they can spontaneously turn on and turn off. So this is just straight out of the chemistry textbooks. It is actually a very reasonably well understood property of how molecules react when you take them down to small volumes. Just to give you a sense of this, if you take the number of free calcium ions in a typical dendritic spine, the number of free calcium ions is of the order of 5 or 6. It's a really small number. Yeah. So you can imagine if you have just 5 or 6, the chances of them doing anything deterministic, yeah, they're going to do things in a very probabilistic manner, stochastic manner. Okay. I hope that answers your question.
So there is one request from Ashutosh Lauter. He has asked that if you don't mind, could you please repeat in brief regarding the associative learning on how the memory is actually preserved longer than the molecules themselves?
Sure. So what happens is sort of summarized in this slide. Yeah, you have a feedback loop, a bistable switch, right? And the bistable switch, if you remember, was one that can be stably in one state or another state. Yeah. So the chemical system will either prefer to be in a state of low activity, say, or a state of high activity. Now, the problem is this chemical system is composed of molecules, and the molecules degrade. And so they degraded here, and now you replace them with fresh molecules. Okay, so it's like taking a reaction system and diluting it a little bit. Now, let's go back to this analogy. So, fine, we were in this state, we removed some molecules, we diluted it a little bit. That is like a small perturbation. That's like a small change to the system. And so the system restores itself to the steady state, state 1. Yeah? Or if it were in state 2, a small perturbation, a bit of dilution, doesn't matter, it will come back to where it was. It takes a very strong stimulus to cause it to change state. In this case, the way you can think of it is that the blue molecules are, let's say, they're not phosphorylated. The light blue molecules are not phosphorylated, the dark blue molecules are. And so the dark blue molecules are active and include some enzymes that cause phosphorylation of these. And so these are brought into the same state as the majority molecules which were left over from the earlier degradation step. And so then the new molecules are now also part of the memory. So that's the general idea, that you bring in new molecules and then you convert them into the same state as the majority of the molecules there by this feedback process. I hope that explains it.
The next question is by Anuthi Tiwari. They ask that, is the memorization power genetic, or can we really train our brain to get better at creating memories and remembering things?
So, of course, there's genetics to everything. There's also a lot of environment to everything, right? You know this. So, yes, you can train yourself. You can, the sensible thing to do is to find environments which allow you to focus. If you want to learn something well, it's a good thing not to be too tired or sleepy. There's various stimulants that one can use, some of which are perfectly reasonable, like coffee, which will wake you up a bit, and you'll learn something better. And there are certain stimulants which are a very bad idea, because if you alter brain chemistry in a serious way, you can cause a lot of other damage, including addiction. So there are many ways in which you can alter the capacity for forming and losing memories. But if you're asking this from the point of view of studying, there are just a couple of things. Do it when you're wide awake. Do it when you're, reasonably well fed. Not just after a meal though, because that tends to make you sleepy. And a third one, which I'm sure you're familiar with, which is you repeat it. You repeat the memorization process. That brings in more synapses into storing this information, and therefore you learn it better. So there's nothing new or magic in all of this. It's stuff that you already knew. That's how you have to do it.
The next question is Gati Krishna Mishra. They ask that, sir, nowadays cases of neurodegenerative disorders are increasing, maybe due to genetic changes or old age dementia happens. Sir, in that case, my question is, can we use artificial neural networks to preserve our memories, kind of how we safeguard our precious photos in hard disk? If yes, can you explain it a bit? How exactly can we do that?
Well, you're going into serious future gazing or science fiction even. So, it's, in principle, you could help to, so let me just give you a glimpse of this from the biology side of it first. Memory loss, memory degradation, Alzheimer's and so on, you have to be aware that you're losing neurons throughout your life. And what is remarkable is how robust the brain is, despite this loss of neurons, this loss of synapses. It's only when it crosses a huge fraction, something of the order of 50% of your cells, that you actually start to have real deficits. So the brain is extremely plastic. So what can you do to postpone this? What can you do to prevent it? One, of course, is to not do stupid things like consume large amounts of alcohol or other things that will kill your brain cells. Another converse thing is, if you do exercise, both physical and mental, if you keep yourself active physically and mentally, that is good for your brain. It increases the likelihood that your cells will keep going. The possibility of recording and re-stimulating, well, we're already making baby steps on that, right? I'm sure you've heard of different kinds of brain implants. Deep brain stimulation is used, for example, in Parkinson's and several other diseases now. Cochlear implants have been around for a while. So you can do this, yeah? But to go so far as recovering original memories, that is very, very, very far in the future. It's something that people are thinking about. I think about it too. But it's not something that I expect will happen anytime soon. Just think of the problem, right? You have of the order of 10 to the 11 or so neurons in your brain. You have of the order of 10 to the 14 synapses. How are you going to read out what they all do? So that's why it's a very big challenge. But it's a very interesting question you've asked.
The next question is by David. Are there differences at molecular level for different types of memory, like memory related to some smell or to some sound?
That's a great question. So memories are stored in many different parts of the brain. And even to zero order, you can make the case that, yes, there are some differences in the receptors, in the protein composition in different parts of the brain. So to that extent, yes. But when it comes to the hippocampus, right, which is kind of an integrative area which associates all sorts of sensory input, all sorts of even contextual input, like whether a previous action led to a reward. All of these are part of forming a memory, and they all happen in the same cells, the same synapses. So it's not that there's a particular memory signature of a kind of sensory input when it comes to a given brain region. It's just that different brain regions tend to specialize a little bit in different kinds of sensory input. So to that extent, there may be some specialization at a molecular level, but not, for example, in a general region like the hippocampus or certain regions of the cortex. Next question.
Okay, so the next question is, as you mentioned that we can look for memory at several levels of organization in the brain, do systems at higher levels emerge due to the activities at the lower levels, analogous to how cells emerge from interactions of organelles? This is from Manas Raikar again.
Again, that's a thoughtful question. So I would like to put it this way, that these are part of an integrated whole. It's not that information or events are flowing upward to more and more abstract levels. It is actually really very much an interplay. Yeah? The circuit, the activity of circuits, in fact, of behavior, will cause events to take place at the molecular level and vice versa. So it's not a one-way street. It is very much the whole brain at all its levels is continuously exchanging information across levels as part of its function and as part of learning.
So the next question is about another theory. What if a person who is unable to create new memories, like the HM case, and they keep a record, like a diary or journal entry of all that is happening? Will the person be able to remember that all of this happened, or will it appear to them like some sort of deja vu or anything else?
Sadly, no, they wouldn't even remember that they made a diary. I mean, they could be told that they made a diary and they could read it, but it would be completely like a story that someone else has written. So, patient HM, as he got old, he had his memories of being a young man in his 20s. Yeah? And here he suddenly was in an old man's body. It was very, very difficult for him to experience this. So yes, people could explain to him, and they would have to explain to him every time that this has happened. But he never really was able to anchor himself to any point in time. It would be like reading a story that happened to somebody else. Yeah. So yes, you could maintain a journal entry, but it's not the same thing as it happening to you, as it being part of what you think of as your experiences.
The next question is by Achinta Kumar Jena. Referring to the two-photon image, how do you confirm that all the active cells do participate in the memory formation process?
No, they don't. In fact, I had a slide here. I'm afraid I went through it a bit too quickly. Here, this image over here. So the cells in red are not participating in, as far as we can see, any of these particular memories. It's the other colored cells which are participating. So a fraction of cells are only participating.
Next question is by Manu Kumar. Patterns and procedures can be stored in the form of circuits and connection, but what about storing independent bits of information? Is there always association?
Okay, I'm not sure I quite follow that. Specific bits of information, if you're referring to as facts that you have read, they too are patterns of input of some sensory form or the other. They too are associations between what you may have learned about, some rule of algebra and therefore its corollary. So they too are inputs which are in context, which involve many, many other kinds of information. Storing something as a bit on its own is not really what brains do. It's the kind of thing you might do on a computer. You might set one bit of memory to one thing or another. But the brain, one of the reasons why the brain is so good at associative recall and these things is because everything is stored in context.
The next question is by Shubham Jyotinanda. He asked that in the case of a disorder where one has the inability to forget, till what point can the brain store memories after which the memories will become a burden?
Well, you should simply read the book that I mentioned to you, The Mind of a Mnemonist. They were not able to find a limit to the duration, and the capacity of his memory seemed to be pretty much boundless, but it was limited because he took a little bit of time to store the information. He had to spend little time sort of consolidating things, relating them. He has certain tricks of how he would structure things in for example, in a imaginary path that he would walk. So he took a little time to store these things, but once he stored it, it seemed to go on indefinitely. Yeah. So I mean, but I think one shouldn't get too hung up on capacity. We have 10 to the 14 or so synapses, but nowadays you get data storage arrays with much larger capacity. So it used to be that it was an incredibly big number. That's when I was a graduate student. But now you guys, you're in a data-rich environment way beyond the capacity of the brain. It's a sobering thought. So maybe we should start to wind up. Maybe just one or two more questions.
Yes, sir. Then I will take 2 or 3 more questions, and then we can just document the questions and send to by mail if possible. So the next question is by Avigyan Dasgupta. They ask that, sir, a recent article claims that there might be a connection between the COVID-19 viral attack and a potential Parkinson's disease. How can this be explained?
Well, many viruses are known to infect neurons. And in fact, COVID-19 is one of them, right? One of the symptoms of at least some of the variants of COVID-19 are loss of sense of smell. And that appears to be because it goes in and affects and possibly even kills the neurons. Now, it's conceivable, I don't know of any mechanism by which they could reach the substantia nigra or other parts of the brain which have which are implicated in Parkinson's. It's conceivable that it could get there. Some viruses like rabies virus are very good at jumping across synapses and therefore getting deep into the brain. I'm not aware that COVID does this. So I will leave that, something that you should do a little research on. There's so much work going on COVID that I certainly have not kept track of this particular thing. It's possible, but I don't know how it might be. So I have to claim ignorance there. Next question.
Next question is by Soham. Are dreams the expressions of the processes of transfer of memory from the hippocampus to the cortex?
Oh, very good. You have been reading some neuroscience. So this has been one of the ideas in the field, a major idea in the field. In fact, one of my lab mate, Matt Wilson, was instrumental in doing some of those experiments of replay many years ago when he was working with Bruce McNaughton and since then in his own lab at MIT. So it's one of the major hypotheses in the field that you use the replay of events during dream, which can be quite muddled, as part of reinforcing memory. I'm sure there's much more to it than that. And there are other theories also. So it's one of the theories though. Okay, last question.
The last question, the brain functions primarily by chemical and electromagnetic signals. Is it possible to be able to read a person's thoughts using implants that can record electromagnetic fluctuations? If so, can we also write information on the brain?
Okay, so that's 2 questions. Can you read person's thoughts? You can read some very simple things. For example, there are certain potentials which you can read even using EEG, which signify that you're about to perform some action, right? So some muscle events have as preliminaries certain brain activity, and you can do that. So about 1/3 of a second before you actually do something, you can detect that this is going to happen. Reading thoughts is a much, much more nasty, complicated process. And I refer you to an earlier question where you were asking about whether you could store the information that was there in the human brain. You have 10 to the 11 neurons and 10 to the 14 connections. How much of that are you going to be able to sample? Very, very little. So the ability to read thoughts and so on is limited hugely by the ability to record from any decent number of neurons. Now, the second part of it was stimulation. Yeah? How do you trigger activity in neurons? And again, a similar issue applies that in order to trigger activity, which you can do, for example, cochlear implants are doing exactly that. They are converting sound waves into electrical signals which trigger neurons. you can do that. It's routine. And you can do it at a reasonably large scale now in experiments, up to 700 neurons. Yeah? But 700 neurons, as opposed to a few billion, is a very, very small sample. So there's a huge limit to what you can do. If you really want to stimulate a large number of neurons in an organized, effective way, I recommend to you to give them a piece of paper with an image on it. Through millions of years of evolution, your eyes, for that matter, your other senses, have evolved to convey information into the brain where it is processed very efficiently and can affect your behavior. Remarkable, isn't it? So you have a built-in stimulation system for triggering activity in the brain. Why don't you use that? Okay, maybe we can wrap up now.
Yes, sir. I think we should wrap up now. It has been a long meeting right now, and there are some few more questions in the chat. We will document them together and probably send you for, if possible, if you agree with that.
That's fine. That's fine. Thank you so much for a very, very lively and very engaged discussion, and good luck to all of you.